The only reason the word bumping uglies is up here is because there's multiple different types of planarians. There are planarians that split and it's asexual reproduction. There are other species of planarian that have sexual reproduction. What did this paper show? What the question was here is, when we age, what are all the bad things that happen when we age, right? When we age, you lose neurons in your brain. The cutaway gag from Family Guy, where there is one neuron left and then he trips over and he breaks his reading glasses and he can't read anymore because everyone else has died and now he doesn't have his glasses. That's kind of true. You do lose neurons as you age. You also lose muscle mass as you age. You're also less fertile as you age. Also, if you get a cut when you're old, it's harder to heal yourself. So how can we fix this? It's the $1 million question is, how can we fix aging? So other research, and the data is cited here as well, they've looked at ways of turning back the biological clock in particular tissues, and how does that happen? But here, and other studies, they also want to do like age reversal of blood cells. So taking blood cells and turning those back earlier in terms of time. Here they looked at planarians, and they looked at a sexually active type of planarian that is called Schmididae mediterranea. Bonus points for anyone who can tell me where it's from. Schmididae mediterranea. And so they're interested in planarians, right? Because they can be considered as immortal, and they can grow new heads when they're decapitated, and new tails, all the things, right? They do still have an aging effect. So if you don't cut them or cause them to actually undergo some level of reproduction, or like in terms of like regeneration, about after 18 months, they have gone like, they're showing signs of aging. So after 18 months of life, from like being a fertilized egg all the way to that, 18 months like being a grown-up little worm, they have loss of neurons. They have loss of muscle cells. They are less fertile, right? That is the same thing that happens with mammals. Their eyes look funny. Here you go. So these are what the eyes look like. This is a young one. They look like cartoon characters, right? They look like those derpy, googly eyes. This is the young one. Those are the old ones of one species. And then here is the other. This is the species that we've seen a lot. Actually, maybe we've seen both, because there are other strains of Mediterranean as well, or the Schistomina, not just the Mediterranean one. But you can see with this one too, in young eyes, and here's older. So you start to see these errors, right? Ectopic eye means that there's a one eye that's not in the right zone. There's just this random eye cell hanging out. Here, the actual pupa has duplicated and it's within the eye itself. Here, the whole eye has duplicated. So those are visual representations of what's going wrong. Let me show y'all what the stem cells look like, though. There was a figure in this paper that was a supplementary video, and it highlights these stem cells. So there is the planarian. Can y'all make out the eyes? I imagine it's straightforward here. Right down here at the beginning of the video, there and there. Those are the eye cells right there. But so those bottom are the eyes, and everything in purple, those purple dots are the stem cells. And so what this video is showing is it is cycling through the animal. So it is starting up at the top and is going through the bottom there. What do you mean there? So that's just the orientation. So it's swimming around Peachka, and they've chopped off the top. So they took a cutting and cut here. They've cut here. That's all regenerating. But see, as you're going through the sample, all those pink dots are stem cells. So that's your stem cell population. There's stem cells all throughout this animal. And why that's important is they somehow know what to become when an injury is triggered. And so, like, that's the $1 million question is, okay, how do they know what to become? How do they know when they've completed becoming whatever cell they've needed to? And then how do they know that it's time to stop dividing and not continue on? Because if they continue on, it is a great way to just destroy the organism. What do they use to illuminate it? So there's a couple of different things, Jay. There are antibodies that work in planarians that mark neurons. So on the outside of the neuron, there is a protein that is like a neuronal specific protein. There's also an antibody like that for stem cells. There's a marker outside of the stem cell that's specific, this protein that only exists on stem cells and doesn't exist elsewhere in the other cell types. So you can actually do stains that have multiple different components in terms of labeling. which again is a really, really powerful tool to be able to utilize. And then you can mark a lot of things in the organism. There's also, if you put it, what if you put it in a blender, injected it where, Peachka? In theory, if you do a cutting and it has stem cells in it, it is sufficient to regenerate. So the way that it is reproduced in the lab is a razor blade that you do a lot of different cuttings on there. Oh yeah, no, the stem cells are not good. Like if you inject some of these stem cells in there, no, like your body would recognize it as foreign. Because so Peachka, the way the immune system would be working is there's cells that are basically policing your bloodstream, and as they're policing your bloodstream, they're looking for cells that have chemical markers of them on them that say no, this doesn't belong in here. And so your cells don't have that marker because they belong to you, but the outside of something foreign is identified as foreign because they have a different set of like flags basically on the outside of the cell. And so if they're able to see that, then your immune cells are like, oh, then it's time to nerf this, pretty much. The cells from the entire system. Yep, exactly. Yep, exactly, Maurice. Exactly. I just wanted to show you all what it looks like. So Jay, the components of embryonic staining, organismal staining, there is an ingredient that you have to add called Triton X, and that is a detergent that opens up some of those pores in the cells. Doing that to a living animal would cause extreme stress, and there would be a lot of issues with it. So that ends up, right, a negative effector. So you have to, what is called, fix the animal, meaning put it into like formaldehyde, any other tissue that we talked about, like preserving the brain. Exactly. Fix first, then stain. Fix first, then stain. There are some stains, Jay, that are called vital dyes, and those can be run on a living animal. But those are not the norm. Those are much more the rarity. Those are much more the rarity within this system. And the reason for that is because a lot of the dyes, they're crossing the cell barrier. Upon crossing the cell barrier, they need to like bind to something. If they bind to something, that could mess up the function of whatever they're binding to, and so on and so forth. So that's one of those takeaways there. Your insights for possible for lab time. Yeah. So that exactly, Morris. So that would be histology. And histological samples are also fixed in like formaldehyde or methanol or something, but it's just thin sections for the histologist. And then they stain them with something, right? In this instance, it's going at a similar thing. It's still asking a question of like, what does this look like? But usually you're guided by an antibody stain. So you're marking a certain protein of interest, versus the histologist is looking usually for just structure, right? Like, here I've taken this cross section. Does this look right? It doesn't always look right, and that's your information of if something's bad or not. Hopefully that makes sense. So the end game, Peach, is to get something like this to work in humans, but the process wouldn't be to inject a Planarian into you. The process is to first understand what the genetic components are that lead to this effect, right? How does this end up working? And why does it end up working in a Planarian? What are the genes that are controlling it? And then the idea is, if you had a dish of human cells now, if you manipulated those same genes, maybe you can get the same result. Except now, instead of regrowing a Planarian, you can regrow neuronal connections, right? Which would be a game changer if you can regrow neural connections, right? Because imagine if you have a severed nerve cord, right? The way that you can get past something like that is if you can like totally like regrow them back together. Yeah, or liver or any tissue, right? There are other animals that are being studied for regenerative abilities, like certain salamanders, right? And that's how they're saying, so this is why it can work. If there are similar genetic components that we have in our bodies as the Planarians do, then it's not that we don't have the genetic information, it's just we don't have the programming to turn something back on and then turn it back off after it's regenerated, right? Those are the critical steps to be looking at. So what they're doing here, Jay, they're cleaning the tank. That's the cheap way of doing this. That's a piece of Tupperware, and they're taking out water and potential trash and then adding new Planarian water to it, and then they dilute it away. So that's what you're seeing here. Is there any theory about stem cells signals, where it gets turned on from, how it knows to stop, any indication where it starts? There's some, Jay, there like are some signals, but the catch is knowing where the signals are coming from. So like if you have a signal, like this is the stem cell, how far away does that signal start, right? Like is it a domino effect that goes 10 steps out? And how many steps in of that domino step are we aware of? So that's the difficult part. A lot of it's been studied in the context of also cancer, because in cancer, right, essentially you just have the growth stage turned on and just never turned off. So you've got a little bit of back and forth between the two that you're able to process. But how exactly the system works, it is still very much in its infancy, Jay. A lot of it too is due to difficulties of not being able to work with stem cells for many decades. There are now ways in which you can revert cells to other states. So like you can take skin cells and revert them to a stem cell like state, and now you can study a stem cell like state in a skin cell, but it's not a true stem cell, it's just kind of a stem cell. How quickly the brain works? Nope. So Peachka, chemical signals go at different rates than brain signal, and so it's not even controlled by the brain signal. The way the stem cell knows what to do has nothing to do with the nervous system, right? It has to do with other chemical signals at a localized point of injury, let's say, that then tells the stem cells to activate. Not coming from the brain. Those are localized signals, possibly due to chemical gradients. So that's how the stem cells know what to end up becoming. Stem cells also self-regenerate. So when a stem cell divides, so this we know it too, at least, Jay. The stem cell divides, one of those two cells stays a stem cell, and the other one starts to go off on its merry way in terms of differentiation, in terms of becoming whatever it needs to become at the end of the day. And the big catch between those two components is if the stem cell is not maintained when it ends up happening. Because there are instances where the stem cell is not maintained, especially when you start aging. So it's, again, you have the cell, it divides, one cell stays a stem cell, the other cell starts in its merry way to become something differentiated. But now you need to have signals that tell this cell either to divide again or to stay. And then the other cell that's become something, you have to give it now signals to continue becoming that thing. It's not just like it's divided and then it immediately knows to become a neuron and you can just let it go. There's constant signals coming on in that say, okay, now you must go in this pathway. So it's two paths really that we're studying at the same time. One, right, is coming on in and having the new cell become what it needs to be, which is different based on what cell type you need it to be. The other one is that it's the maintenance of the existing stem cell and either telling it to divide again or telling it to now stay put. Or if you tell it to divide, and here's where it gets even more confusing, Jay, like even in the Planarian, that stem cell is told, hey, it's time to divide again. But maybe this time when it divides again, it doesn't become the same kind of cell, it becomes a different cell. Well, then what, right? So like, it's like multiple signals could be acting here. And those multiple signals could be like, how do you prevent it when you're studying it from getting cross-signaled even? Especially if you want to understand it in the body of the organism that you're looking at, that's not going to be straightforward. Yeah, exactly. There is a high level of complexity, which is important to note that is going on, and that should be accounted for and addressed as well. And it's also just not straightforward. Really cool, not straightforward. So what exactly was done in this paper? So I told you all they were looking at the eyes. Right, the eyes, here we go. The eyes changed as the animals aged, right? So even though they're considered to be like maybe they should be immortal, there is still this aging effect that you can observe. And when you observe the aging effect, you see how the eyes are changing. And so it's like they no longer look like this reliable, here's this two eye set, but rather you've got like multiple dots in there, a doubling, a dot going elsewhere, all these things going wrong. So their question was, and this is a pretty neat question, right, is how does regeneration affect the aging process? So if you just let them sexually reproduce, for about 18 months, they show these obvious, like really strong signs of decline. So what they did was they cut off some heads. And what they find is if you cut off the heads, then not only are the neurons younger in terms of their biological clock, but other parts of their body are also regenerating. So it seems like if you trigger regeneration in one zone of the body, it actually goes into the rest of the body as well. So their fertility gets better after they've regenerated back that head, right? Which is weird because they're only cutting off the heads. They're looking at these eyes look goofy. If I chop off the head, head grows back, and the eyes no longer look goofy. Like, that all makes sense. Like, that's a straightforward hit and a half, right? Like, yes, they should make and look that way. Exactly, Jay. So it seems like it's a stimulus for the entire body of this organism. So what they found is that the planarians don't lose these adult stem cells as they get older. And in fact, if you stimulate those stem cells, that regeneration reversed aged the transcriptional state of a lot of tissues. Now, what does that mean? When a tissue has gotten old, it has certain genes that are turned on and off. And those are called molecular markers of what is like an old tissue. So in old tissues, there are certain things that are turned up and there are certain things that are turned down that are different from young tissues, right? It seems like when you do this cut and you stimulate regeneration, the other organs then revert to the genetic state as when they were younger, which is really, really cool. So the old planarians didn't lose the ability to lose that regeneration. They can still regenerate, which is what a lot of species like can't actually do in terms when there's regenerations that are looked at. But here, again, what was really, really cool was that everything reverted to this younger state. And that younger state reversion is really, really critical that they're highlighting. Like all of a sudden, biologically, everything looks younger. And what's really then cool is, Jay, they wanted to apply this to mammals. Does this biological concept work in a mammalian model? Or are we just totally at a loss and this doesn't work in mammals and this is just a weird planarian thing? So what they looked at was they had existing data sets that they can compare it to. So what they got was they did single cell sequencing from planarians. Single cell sequencing we've talked about is when you're sequencing the RNA levels of just a single planarian and in each individual cell of that planarian to figure out what exactly is different in every single cell. So you can go from the old animal, young animal, and then an animal that has undergone regeneration. And you can see every single cell and how it's behaving in that instant. You can then compare that data set to mammals, right? There are mammals data sets, mice, rats, humans that have undergone aging. And then there's also data sets of mice that have undergone life extending treatments. Okay. So now you're wanting to look at it for that similarity. So if you have a life extending treatment in a mammal, for example, I just said the word antioxidants or caloric restriction. Both of those are known to extend lifespan in mammals, flies, pretty much everything. Look for genetic markers and see if they look similar. And here's the catch. Thank you, Osgoober. Appreciate you. Here's the catch. They actually found the similarity. So it turns out that the genetic signature of the aging planarians were shared with aging mammals. They also found that the planarians that underwent regeneration had genetic matches with the life extended mice, like in terms of the data set. There were a lot of similarities showing you that it is in fact a reversal of the age state. And so why this is important is it's not even yet looking at how to reverse aging, but it's just looking at and figuring out how does that age associated decline happen on the organismal level. And it seems like it might be conserved, right? That a lot of organisms age in similar ways. The genetics behind aging might be very, very similar. And so that's all the more evidence to if you're studying aging in this animal, there will be translational use for it, especially given that mice and planarians are separated by an absurd number of millions of years of evolution. And yet they're sharing similar genetic signatures of intervention and not intervention as well. It is really, really cool. The whole animal of the mammal or site specific. So this was whole animal that they looked at for the sequencing. For the other animal data sets, Jay, they were not single cell sequencing. They were called bulk sequencing. So in that bulk sequencing data set, they did look at certain tissues and they also looked at the whole animal as a whole. And both of those showed clusters of genes chemically similar and acting between the different zones, which is really, really exciting, I feel, right? So either if you looked at it in bulk or you looked at tissue specific, you got matches to the single cell data set that was in the planarian for both aging and for both the intervention as well. Again, the mammal sets were not single cell. Those were, so it was like, I guess, Jay, think of it like I took tissue from the animal, like here's a brain specimen. I'm not looking at what every cell is doing independently. I'm looking at what the organ of the brain is doing as a whole in the brain of the animal. And that's like the easiest, that's the cheapest and easiest way to do it. The justification for doing it like this in the Planarian with single cell is because there are zones, right, where there are regeneration happening, or maybe regeneration is just finished, and there's zones where there was no regeneration happening. So you've got kind of a signaling gradient. And this way you can take a look at that signaling gradient and see, okay, what zone is important, what zone is not important, especially for this behavior that you're trying to observe. So you're just getting a higher resolution of data, and you're able to make that claim to it. So it is I think it's a really, really cool data set that they were able to pull out here. And it's a really neat finding that you're able to have this reverse aging essentially by just stimulating those cells. Now, what the cell signal is during the stimulation part of it, like, can you, one of the next questions they wanted to ask, and they said in the discussion, how do I go in and stimulate this younger state without chopping the animal, right? Without chopping the animal. How can I trigger this? If you figure that out, then maybe that's the way that you can translate it to other animals in a much more easier way, where you don't have to, Jay, like you said, cut your head off. That would be ideal, right? That would be ideal if you did not have to chop off your head each time, or really any time for that matter. It would be nice to just be like, oh, this is my head, this is what it does, and I don't have to worry about dropping any of this. That would be a nicer, kinder, calmer way to do. And that's mostly the paper here. I think it was really well done. Here they also did a variety of the chopping. So in terms of the other data that they got here, they looked at a variety of different chops to show that there were similar features. So this is a really nice built-in control. They even like did this, this cut, and you can see like here's the old eye and there's the new eye. And instead of having old eye appearance with the new eye looks like a young eye. Like, they did a lot of combinations here to make this truly look like it's not just a one-off situation. It's not just a zonal question of how it's done. Like, it is a very unique way of doing this experiment. And it is, in fact, a real thing. Which, again, I feel like is important to do. And if they hadn't done this, I feel like a lot of us would be asking, like, follow-up questions. Like, well, this doesn't quite make sense. And there's other kinds of controls that maybe should have been added in. But with this, it's like, all right, this makes easy. At least any stimulus insofar as the cut. The cut site isn't dependent on you cutting off the entire head or like a zone of the head. It's like, no, we're pretty on point, and it's looking pretty decent. So again, I thought that was like a really nice, easy takeaway for how the system worked. And they even have nice diagrams to show you exactly where they did the experiment cuts. Again, all this will be up on the Discord, as we always have. They have a nice outline of the amputation, regeneration, again with the sexual reproduction to see how it's inherited. They did some genetic manipulation as well, and they did find a few genes that are involved in this pluripotent state. And so they have some immunohistochemistry where they're looking at certain target genes. I do want to share a last thing before we raid out for the evening, just a video on how they do the genetic manipulation, because I feel like that's one of those things where it's not straightforward of how you do it with a Planarian. So here's an example video of the process. So you do an injection into the Planarian, and you inject a genetic modifier. Here's your example injection. So there's the Planarian. There's a little needle. Put the needle into the Planarian. There goes your liquid. I just thought that was a neat bit of, again, just tech to see how the system works. I feel like it's not always straightforward to see how this system works. And so it's just, I don't know, I think it's kind of neat to be able to see, again, like how the takeaway works like that. Back meat fluid, if I can have both shoulder blades removed after I have have those grow back, oh goodness gracious me, that would be so much better. And I just don't have to like live in pain actively anymore. That would be quite lovely. But yeah, they were able to do some genetic manipulations, and they found like with that sequencing data and some of those markers, one thing that they could do, Jay, which I thought was really cool, is they could activate some of these genes without cutting the animal. And they found that like helped maintain this younger state that the animal didn't have before. So by finding your key genetic targets, if you manipulate those key genetic targets in the absence of the cut or the absence of that regenerative stimulus, they were able to get some of it to start working. Some of it started being like, oh, okay, now I have this system working. Awesome. But it wasn't to the same level as before, so there are still some signals missing. Yeah, so they injected the animal to manipulate the DNA or the RNA levels, in fact, to make it so that you lower signals of aging. You lower the chemical signals of what are aging, so you adjust the system back to being something like what they were prior to when the system, like before the age system, right? So you do that, and then it ended up like, yeah, it did. The animal wasn't at the same age state, so it wasn't the injection that triggered it. It was what was being injected. It didn't look quite as good as the regeneration components. There's still probably more things that need to be modified, but it was still really, really cool to be able to have that delivery system. That injection's just been established in the field. That's just like the video that we just watched. That's their standard looking for like how you do the genetic manipulation. And so that's how you know how to do it. It's just like, oh, that's the standard for what we have. And yeah, excellent, if you will. All right, guys, I think that's where we'll leave it for this evening.